My name's Gabby.
And my name's Ashley.
And we're going to be doing the cell cycle today.
Okay, so the first stage of the cell cycle starts off with interphase, and it combines G1, S, and G2. Interphase is usually, where the cell typically spends most of its life, and the cell begins to grow and perform its normal functions. And it also prepares the cell to go into division for all of the stages.
Okay, so G1 is the first of the four phases. The cell grows larger physically, and it makes organelles, and it has the molecular building blocks, and it's also the first step for all these. It's the starting step for all the later steps to come.
Okay, and for the S phase, it's basically more of like a DNA replication, and it synthesizes the DNA by processing all the nucleotides that it has to make sure that every single DNA is replicated, so that there consists of two different chromatids for the cell.
And G2 or GAP2 is the stage where it's starting to grow more and more right before it gets to division, which is mitosis. So the cell right here is already starting to make proteins and organelles. It begins to recognize its contents for preparation of mitosis. And whenever this ends, that's when mitosis begins. And this is this is mitosis and the stages. So it's prophase, metaphase, anaphase, telophase, and cytokinesis. So interphase is just when the cell is barely starting out with the nuclear envelope is starting to dissipate. And here is when the spindle fibers are already forming, and the nucleus is already starting up with the sister chromatids in the middle. Metaphase is when the chromosomes become attached to the spindle fibers, and they line up along the metaphase plate. Anaphase is when the two sister chromatids split, so that's the little V shape right here, and they go to the opposite sides of the poles, the chromosomes. Telophase is when the cell is finally starting to split, and the cells move to each different side of the cells, which looks like a weird 8. And cytokinesis is when it finally separates into two identical cells, or two daughter cells.
Okay, and meiosis is the similar format as mitosis, but instead of only producing two, it produces 4, and they're unidentical to the parents and to each other. And then in the first, one of the stages is crossing over. So it starts off like this, 2 separate chromosomes, and then they pile up on each other. And then this one comes out with the tips a whole different color because they crossed over. And then this one as well changes colors. And that's the process of crossing over.
Okay, and lastly, I'll be talking about the different checkpoints. The spindle checkpoint is made to make sure that the spindles don't separate before anaphase occurs, and it's also to make sure that the chromosomes don't misalign. And for the G1S checkpoint, it just basically checks for damaged DNA so that way it can go on to the replication and division. And for G2M checkpoint, it also makes sure that the DNA is replicated and it's not damaged. And also, if this checkpoint notices like a failure or something, then that's how cancer is made, because the cells keep on forming and forming until it's just a lot. That's pretty much it.
And that's a cell cycle.